Hello and welcome to some biology class. Friends, today we shall discuss about alternative splicing. Before that, I request all of my friends, please subscribe my YouTube channel. Also watch, like and share my videos with your friends. Splicing is a biological process that removes the non-coding sequences or introns from a pre-mRNA and joins the coding sequences or exons together to produce a functional mRNA molecule. It plays a central role in the processing of pre-mRNA and thereby open the possibility of the regulation of the gene expression by control of the splicing machinery. Since most pre-mRNAs contain multiple introns, different mRNAs can be produced from the same gene by different combinations of 5' and 3' splice sites. This possibility of joining exons in varied combinations provide a novel means of controlling gene expression by generating multiple mRNAs, therefore multiple proteins from the same pre-mRNA. This process is known as alternative splicing, which occurs frequently in the genes of higher eukaryotes. For example, about 50% of human genes produce transcripts that are alternatively spliced. This process considerably increases the diversity of proteins that can be encoded by the estimated 20,000 to 25,000 genes in the mammalian genome. Because the pattern of alternative splicing vary in different tissues and in response to extracellular signals, this process provides an important mechanism of the tissue-specific and developmental regulation of the gene expression. One well-studied example of tissue-specific alternative splicing is provided by sex determination in Drosophila. In this organism, alternative splicing of the same pre-mRNA determines whether a fly is a male or a female. The pre-mRNA is transcribed from a gene called transformer. The transformer pre-mRNA contains 3 exons, out of which the second exon contains a termination codon UAG. The arrow marks indicate various splice sites. In male Drosophila, exon 1 is joined to the most upstream 3' splice site, which is recognized by a splicing factor called U2AF. Similarly, these two splice sites are also joined. In this case, the second exon, which is included in the mRNA, contains the termination codon UAG. Because of this, its translation is prematurely terminated and no functional proteins are produced. In case of female Drosophila, the situation is different. In this case, a repressor protein called SXL binds to the most upstream 3' splice site. By doing so, it blocks the binding of the splicing factor U2AF. SXL protein is only expressed in female flies. Because of this, a part of the second exon that contains the termination codon is skipped in mRNA. In female flies, the splicing factor U2AF recognizes the alternate 3' splice site which is located further downstream in the second exon. So exon 1 is joined to the alternate 3' splice site, which is located further downstream in the second exon. Because of this, the part of the second exon which contains the termination codon is not included in the mRNA. Due to the absence of the termination codon in the second exon, in female flies, the transformer mRNA is fully translated and functional transformer proteins are produced that act as a key regulator of sex determination. So in this way, the same transformer pre-mRNA is alternatively spliced in Drosophila, and this contributes to the sex determination. The alternative splicing of transformer pre-mRNA illustrates the action of SXL protein that functions by blocking the binding of the splicing factor U2AF. A large group of proteins similarly regulates alternative splicing by binding to silencer sequences in the pre-mRNA. In other cases, alternative splicing is also controlled by splicing activators that recruits splicing factors to sites that would otherwise not be recognized. The best studied splicing activators are the members of the SR protein family, which binds to specific enhancer sequences in the pre-mRNA. Multiple mechanisms thus regulate alternative splicing. Variation in this type of splicing pathway makes a major contribution to the protein diversity during development and differentiation. One more striking example is the alternative splicing of a Drosophila cell surface protein called DSCAM that specify connections between neurons. Friends, this is all about alternative splicing. For more updates, subscribe my YouTube channel, Sam Biology Class.